What is a bacteriophage? A bacteriophage is a type of virus that infects bacteria and uses a host cell machinery to replicate. It consists of a capsid which contains viral DNA, a tail, and a tail fiber. In this video, we will be looking at the lysogenic and lytic cycle of a lambda-phage that has infected an E. coli cell. Before the lysogenic or lytic cycle can begin, the bacteriophage must insert its genetic material into the host cell. Each bacteriophage can only infect a limited number of host cells, which is called the host range, as there must be a lock and key fit between viral surface proteins and cell surface proteins. In this case, the lambda-phage binds to the surface cell receptor responsible for maltodextrin transport. Once attached, the bacteriophage can inject its DNA into the cell. Once the viral DNA is inside the cell, the linear ends of the genome immediately come together to form a circular structure.
Following the formation of the circular phage DNA, the lambda phage can go into either the lytic or lysogenic cycle. First, we will explain the lysogenic cycle. On the lambda phage DNA, there is an ATTP site. On the bacterial genome, in this case on the Escherichia coli genome, there is a corresponding ATTB site. The P in ATTP stands for phage and the B in ATTB stands for bacteria. Furthermore, the lambda DNA transcribes and translates an enzyme called integrase. Integrase hydrolyzes these regions on the phage and bacterial DNA. It uses sticky ends on the bacterial genome to attach to the sticky ends on the phage genome. The phage DNA has now been incorporated into the bacterial genome and will be replicated when the bacteria replicates its DNA. During this lysogenic cycle, the phage DNA will be present in all daughter bacteria cells.
In order to maintain lysogeny, the prophage's lysogenic genes as well as the C1 gene, which is a regulatory gene, are transcribed and translated into proteins. The C1 gene produces C1 proteins, which forms dimers and acts as a repressor that inhibits the transcription of lytic genes. The C1 dimer also promotes the transcription of the C1 gene, thus forming a positive feedback loop where more C1 is produced.
The bacteria may then enter the lytic cycle. Once the E. coli bacteria are present in a favorable condition, the bacteria produces proteases that degrade the highly unsavory C1 gene that inhibits the lytic cycle.
After the C1 dimers are degraded, the lytic genes can be transcribed. One of these genes codes for excisionase, which interacts with integrase to excise the phage DNA from the bacterial DNA. Shortly after, the bacterial DNA is degraded. Once the circular structure of the phage DNA is reformed, genes that code for the phage protein head, tail, and tail fiber are also transcribed and translated. In addition to this, the phage DNA replicates. The new copies of phage DNA are each enveloped by head proteins, which attach to tail proteins and tail fibers. The new phages are all assembled in the bacteria.
Wall phages are being assembled. Proteins that lyse the bacterial membrane and cell wall were produced. These proteins first degrade the cell membrane, then the cell wall. After this process, the phages are released from the bacteria. They can now infect other cells, commencing the lytic or lysogenic cycle once again.